Okay, so for this class, it's going to be loads of exam questions that we're going to go through for biology. This is a mixture of Edexcel and AQA exam questions. And as we're going through the questions, I'll go over the content as well, just as we're going through. So we'll get started because we've got quite a few questions to go through. So this question's about cells. Figure 1 shows an animal cell. Label parts A, B, and C on the figure. So B, you should recognize that as a nucleus. You've got A and B. So A is pointing to the membrane that's around, so that's just a cell membrane that controls what goes in and out of the cell. The nucleus tells that cell what its job is. So the brain of the cell. Okay, and then part C is pointing to the middle, so that's just so remember, animal cells are a type of eukaryotic cells, so they could have given a different eukaryotic cell. You could have also had a plant cell. Plant cells are also a type of eukaryotic cells. Remember, the difference with the plant cells is that plant cells have chloroplasts, they've got a permanent vacuole, and they've also got the cell wall extra. So they've got 3 extra things compared to an animal cell. And it also has everything the same as an animal cell as well. Okay, that's question 1. Figure 1 shows a bacterial cell. So bacterial cells are types of prokaryotic cells. Prokaryotic cells. And the difference between a prokaryotic cell and a eukaryotic cell is that prokaryotic cells don't have a nucleus. They just have their DNA just free floating. So they could have it as a plasmid ring. As you can see here, it's just free floating in the middle. Okay, so it says what is structure W? So you can see W is the extra wall that's around, so that's the cell wall. You can also use process of elimination whenever you're given options. So it's definitely not the cytoplasm, it's definitely not the DNA because the DNA is in the middle. And then D, the plasmid. A plasmid is a DNA ring. Give the name of structure Z. That's the tail. We call that the flagellum because it's just one. Flagella is if you've got multiple. So flagellum. And it says state the function of the structure Z. Well, think about it. It's a bacterial cell, and the whole point of the bacterial cell is a type of pathogen, and it wants to infect. And in order to infect, it has to be able to move, and the flagellum helps it move. So you would just say movement. Whenever you're looking at these exam questions as well, always look at the marks as well. So you can see these are all 1 markers. They don't want you to go into much detail. Just keep it really simple, no waffle, so you just get the mark. Okay, moving on to figure 3 shows a different animal cell. Calculate the magnification of the image in figure 3, completing the following steps. Now, this question is an easier question because they're giving you steps to do this. Had this been on a higher paper, they wouldn't have given any of these steps to actually help you calculate the magnification. They just would have expected you to do it. So it's the triangle that you need to remember. So the I am triangle. They will give you this in the equation sheet. You have to memorize this. So I stands for image size. So that's what you're actually measuring. So what we're going to measure from Y to Z using a ruler. I don't have a ruler, so I'll tell you what they expect that number to be. That's what you're measuring. That's the image size. A is actual size. What's the actual size of that cell in real life? And then M is the magnification. So magnification is how much have you zoomed in to make this image as big as it is. Okay, so it says measure the width of the image from Y to Z. So that's what we start off with because that's going to find our I. So measuring that, you end up getting, if you actually measured it accurately, you would have got 3.6 centimeters or 36 millimeters. It's guiding you to put it into millimeters. So yeah, 3.6 centimeters you would have measured. You times that by 10, and that gives you the 36 millimeters. Or you can see there's even millimeters on your actual ruler as well. So you can see 10 millimeters is 1 centimeter. Okay, so we've just worked out I, so that's our value of I. It says given the width of the image is in, we want to give the actual size in height. And they've given you, oops, 2 seconds. Okay, they've given you the what units to put in, and they've also given you the conversion. This conversion will be given to you if you're doing the higher paper, but for foundation, they do give you it. I have seen instances as well where they don't also give it in foundation as well, so it is worth memorizing that conversion. So it says here 1 millimeter is 1,000 micrometers. We've got 36 millimeters. So if we've got 36 millimeters, to get from 1 to 36, we're timesing by 36. So we would times the other side by 36 as well. So 1,000 divided by, times by 36 gives you 36,000. 36,000. Okay, so we found the actual size now. So we know I I'll just put it over here. We know I in the triangle is 30, well, not 36, sorry, 36,000, because we've times it up to micrometers, right? Your I and A always have to be in the same units. You can't have one of a millimeters and one of a micrometers. You have to divide the same units out when working out magnification. Okay, so now it's giving you the equation for magnification, but if they didn't give you that, again, you can work it out from the triangle. There's your M, so M equals I divided by A. M equals I divided by A, which is what they've given here as well. So magnification is the width of the image, which is I, divided by the real width of the cell in micrometers. So they've told you they both have to be in the same one. And this question's helped you quite a lot. They won't usually do that. They just let you just do it for 4 marks. Okay, so the magnification equals the width of the image in micrometers. So the image size was 36,000 divided by, has it told us? Okay, so yeah, it's told us the real one here. So the cell in figure 3 has a real width of 40 micrometers. So that's what we're dividing by. So divided by 40, in the calculator, you end up getting, okay, so putting that in your calculator, you end up getting 900. So this animal cell has been zoomed in 900 times in order to get this image, essentially. Now, there's one thing I just want to cover, the difference between magnification and resolution, which students tend to get quite confused with. So magnification and resolution. So magnification is how much you make an image larger. So something actual, how much larger you're making it. So think about if you've got your phone screen and you're zooming into something, that's magnification. So it's how much bigger you're making something. Whereas with resolution, so imagine you're zooming into an image and it's getting blurrier the more you zoom in, right? So the resolution is getting lower because you're not able to distinguish between the 2 points. It's just becoming blurry. So for example, if you look at something in front of you right now, you're able to, you've got a high resolution the way you can see right now, because you can distinguish the difference between 2 points. You can see clearly, but someone from a really further distance, someone looking from a plane looking down, they wouldn't be able to see the difference between the 2 points. Everything would just be quite blurry. So that's resolution, the ability to distinguish between 2 points. And this is a really important difference between the light microscopes and the electron microscopes. Now remember, the light microscopes are the ones that you use in school. They just rely on light. They're quite cheap to use. The resolution isn't super high, but they can magnify in. Resolution, the electron microscopes tend to have a higher resolution. But the issue with the electron microscope is you have to have the specimen to be dead. So you can't look at stuff that's alive with the electron microscope. And it's also really expensive as well to use that. So again, that's quite a common question as well. The resolution and magnifications of microscopes and the differences, pros and cons between light microscope and electron microscope. Okay, figure 2 shows a method used to extract DNA from strawberries. So they've given you stages. So let's have a look at the stages. Always understand it before just jumping straight into the question. So stage 1, they've got a beaker, crushed strawberries inside with salt solution and washing up liquid. Stage 2, you can see they're pouring the solution into a funnel, and it looks like there's filter paper there. And what they're doing is they're filtering out the liquid containing the DNA into that tube, and I guess leaving everything else behind. Then adding ice cold ethanol to that solution, and then you've got that mixture. So it says complete the sentences using the words from the box. So crushed strawberries are mixed with washing up liquid, and it says in the question, salt solution. So salt solution. Again, because this is a foundation question, they're prompting you to just read the question. They won't give you this support for higher. So yeah, it's always, whenever you're doing the DNA experiment, they always mix it with washing up liquid and that salt solution. And the washing up liquid essentially helps to release the DNA by breaking open the cell membrane. That's the reason why they add the washing up liquid, breaks the cell membrane in order to get that DNA out, essentially. So describe the method shown in stage 2. Well, we just spoke about what's happening in stage 2. They're pouring the solution into the filter paper in order to filter out and separate the DNA. So let's write something along them lines. You can also write these questions in bullet points just to explicitly state to the examiner that this is where my mark is coming from. So there's a 2 marker. You need 2 points to essentially get the mark. If you end up writing a 3rd point and that 3rd point's not relevant, you lose the mark because because of relevancy. You're just writing everything that you know. Okay, so they're pouring the contents into the funnel. To be specific, where is it going exactly? So you could see it's going into this funnel here. And then you could say inside the funnel, you've got filter paper to essentially separate out the DNA. To separate out the DNA. And then my 2 points, just really explicitly stated for the examiner, 2 marks. Okay, what is the color of the DNA precipitate? The precipitate that comes out at the end with all these DNA experiments is always white. That's something that you do need to remember. Okay, moving on to the next question. So scientists think that chickens were domesticated from the raped jungle fowl thousands of years ago. Figure 4 shows some information about these birds. So they're given a photo of each bird. So you can see the difference is that one earlier on was quite small, and the domesticated chicken today, they're a lot bigger. You've got the mass of them both. So you can see the mass of the first one is a lot lower compared to the one that's around today, and the number of eggs laid is a lot lower compared to the ones today. Which is quite significantly higher now. So describe how selective breeding has produced chickens that lay large numbers of eggs. So we're essentially just describing the selective breeding process, which is the same always. You just need to apply it to the context of the question. So in terms of the question, so I need 3 points because it's a 3 marker. So describe how selective is produced chickens to lay the large number of eggs. So you're going to select birds which essentially have the highest egg production. So select birds that have the highest egg production, because that's the one we're wanting to breed. Okay, so that's your first point with selective breeding. It's always the same format with these selective breeding questions. You're then going to breed these birds together. The ones that have the highest egg production, you're going to breed them together. So you're going to breed them together for your next point. So then once you breed them together, you then select from their offspring which one has them traits. So from the offspring, so the birds that they give birth to, select the birds which have the trait. You could even be more explicit, the trait of laying large number of eggs. And this is done over many generations. So by many generations, so once that them 2 birds have had offspring, you've selected that offspring which has the best eggs. You then keep repeating that cycle over many generations, and that's selective breeding. Selective breeding questions always similar format. Just always make sure you apply it to the question what they're asking for. If you didn't apply it to the concept of large number of eggs, you would have lost the mark, because you have to give it in terms of context. Okay, chickens have 78 chromosomes. You don't have to remember what animals have what amount of chromosomes, but you do have to remember that humans, we've got 46 chromosomes. Or you could say 23 pairs, but if you just say 46, that's really explicit. 23 times 2 is 46 of chromosomes. Okay, so you need to remember the humans one, but if the question's not in terms of humans, they'll always tell you. So for example, here chickens have 78 chromosomes in each of their body cells. Which term describes a chicken body cell? Dominant, no. Haploid, no. Recessive, no. Diploid, yeah. So all normal cells in the body, this for humans as well, are diploid cells. So diploid cells in humans have 46 chromosomes. The haploid cells have half the amount, so they've got 23 chromosomes. And they would be the gametes, so the egg and the sperm. So they're the only ones that are haploid cells, but every other cell is just a normal cell and has 46. So diploid versus haploid. These terms, dominant and recessive, are to do with alleles, which we've got a question on that, so we'll come on to that. It says state the number of chromosomes found in the gametes produced by the chickens. Well, the gametes are just going to have half the amount of that 78, so you're just doing 78 divided by 2, which gives you 39. Yeah, 39 chromosomes in the haploid. So with a haploid, you just half them. Okay. Complete the table to compare the production of body cells and gametes. One box has been done for you. So the body cells, these are essentially the diploid cells. Just keep this as note. The gametes are the haploid, so they have half the amount. So the diploid cells are formed by mitosis. So remember, mitosis, the whole point of mitosis is for growth and repair of normal cells. So the diploid cells, that's done by mitosis, and mitosis produces clones. So whatever it produces is exactly the same as what it started off with, and the number of daughter cells that are produced from there is 2. So that one that you start off with ends up duplicating, and that's 2. The gametes, the type of cell division for that is meiosis. It's really common for students to misspell this, so just be careful. MEIOSIS. Let me write that again. So MEI, meiosis. Okay, so they to produce the haploid cells, which are the gametes. And these cells are all genetically different. So they're not all the same at the end, because you wouldn't want all the gametes to be exactly the same. Like, for example, if all the eggs looked exactly the same, then you would look the same as your siblings. You don't want that. You want that genetic difference. And at the end of that process, you get 4 daughter cells that are produced, because each of them have half the amount with them. So that's why it's 4 rather than the 2. Okay, so some animal cells are stem cells. You need to know where these stem cells are found. So in animals, stem cells are found in the bone marrow. So the bone marrow is essentially that spongy layer that's between the bone, essentially, and they're stem cells. Or you can find stem cells in embryos, but you can see how that raises ethical concerns of using an embryo for that basis. So describe the function of stem cells. Well, stem cells can be differentiated into anything. So okay, so describe the function of stem cells for 2 marks. So you would say the function of the stem cells is essentially to make new cells. You can differentiate into any cell that's needed. So make new cells, and these new cells can essentially differentiate. And differentiate means become a certain cell. So they can differentiate. And what else could you say about this? Some animal cells have stem cells. Describe the function of stem cells. So you're using stem cells to make new cells, and these differentiate to essentially replace damaged cells. But again, they're found in the bone marrow if they ask where it's found. Also, another thing with the bone marrow stem cells, which I'll just keep a note over here. So the bone marrow stem cells, they can't be differentiated into any cell. They can only differentiate into red blood cells. Whereas the embryonic stem cells, they can differentiate into anything. But then it's that ethical concern of using an embryo to develop anything that you're wanting. Okay, moving on. So figure 5 shows different parts of the blood. Now remember, the blood has 4 different things inside it. So the blood is made up of red blood cells, is made up of white blood cells, it's made up of plasma and platelets. You need to know what each of these look like, how they're adapted by what they look like, and what's their job. So red blood cells is to transport oxygen around the body, so that's used for respiration. So let me put notes on that. So there's to transport oxygen. The white blood cells is for the immune system response. So the phagocytes, the lymphocytes, and so on, is to do the immune system. The plasma essentially holds everything together. It's a liquid in between. And then platelets, they clot the blood. The reason why you would want to clot the blood is, say if you get a cut, for example, you don't want to be continuously bleeding out. It scabs. That scab is caused by the platelets. So they're the 4 things that are inside the blood. If you look at this diagram, we've got A, that's probably pointing to the plasma. We've got B, we've got D and C. D is the platelets. It says which letter shows the white blood cell. So the white blood cell is the bigger one. So D's in the middle, C. The other ones, B, they're all red blood cells. They're the concave, small shaped one, and there's loads of them. because they need it for oxygen transport. So yeah, C is the white blood cell that it's pointing to. Which part of the blood causes the blood to clot? That's the platelets, as we just discussed. Name 2 substances transported in the blood. Now, there's so many things that are transported in the blood. We've already spoken about oxygen being transported. If you think about the endocrine system, the insulin and glucose and such, that's all to do with blood glucose concentration. So glucose is also in the blood as well, that gets transported for respiration. But I'll list a few other things as well. So there's hormones. You could have put urea down. You could have put carbon dioxide, because carbon dioxide we breathe out. You could have said water. You could have said amino acids, fatty acids, glycerol, antibodies, which come from white blood cells, lactic acid, minerals, and vitamins. So yeah, there's others that are there as well, but these 2 are your most safe ones, is what I would say: oxygen and glucose. Healthy people can volunteer to donate blood. Okay, the donated blood is then given to the patients who need the blood. When the volunteer donates the blood, the volunteer's health is checked. A needle is used to remove the blood from a vein in the arm. The volunteer rests for 15 minutes after the donation. So there's loads of blood banks around. So you could go, I think you have to be a certain age to go. I think you have to be 18. Because we've all got different blood types and your body will only take a certain blood type. So that's why they constantly need donations from people around. So you go in, they essentially take a few bottles of blood from your arm, but they make sure your blood is in a good condition before doing that. Okay, so these suggest questions, if you ever get a suggest question in the exam, it means that you've not learned this in biology, but they're saying use your knowledge on what you know to kind of answer this question. So yeah, you will get these suggest questions on the paper, unfortunately, where they're just testing you to apply your knowledge. So here it's saying suggest one reason why blood is removed from a vein and not an artery. So what you need to think of is the process of both the vein and the artery. So let's write this down. So we've got artery, we've got vein. Arteries take blood away from the heart. To remember that, it's the A and A. So if arteries take blood away from the heart, then veins take blood to the heart. Now, when the arteries taking blood away from the heart is taking the blood away at a very high pressure. If you think about the aorta, where does the aorta take the blood? It takes the blood to the rest of the body to give the oxygen for respiration. So it has to go at a very high pressure. So you wouldn't want to take blood from an artery because it's under high pressure. If you puncture a hole in something that's got high pressure, it's just going to be quite explosive, right? Whereas with the vein, it's under a lot lower pressure. So that's why it's a lot better to take blood away from the vein than it is the artery. So when they do a blood test or they on your arm, they take it from the vein. So that's what you could have said. You could say blood is under lower pressure in the vein. Another reason why you can remember that as well, which you need to know, remember the veins have valves inside. If I just draw both these, arteries don't have valves inside, but the vein does have a valve inside. The reason why the vein has a valve inside is to keep that blood going in one direction. The artery doesn't need that valve because the blood is going at such a high pressure that it stays in one direction anyway. So that's the reason, another reason why the vein also has a valve, because blood is under lower pressure in the vein. So they wanted you to apply that knowledge to this suggest question. There was other stuff that they accepted in the mark scheme as well. So it says easier to puncture the vein and easier to access the vein, which is probably not as intuitive to write down. It says less painful and blood flows smoothly. Again, I wouldn't expect that to be on every mark scheme, so it's probably safe to go with the pressure answer is what I would say. Okay, question 7. Deposits can form in coronary arteries. Now, coronary arteries, coronary means anything to do with the heart. So coronary arteries, arteries specifically on the heart. Generic arteries are all over. If you see the word pulmonary, pulmonary is to do with lungs. Make sure you understand them 2 words. Coronary to do with the heart, pulmonary is to do with lungs. It says the deposits are called plaques. Plaques is essentially similar thing as when you go to the dentist and they're like, oh, you've got plaque on your teeth, that yellow substance that's building up. It's similar plaque also that's being built up in the arteries as well. So plaques cause damage to the arteries because it's essentially reducing them, right? So you can, yeah. How is this all being recorded? Yeah, this is all being recorded. Yeah, it's being recorded, and I'll send these notes out as well. Okay, yeah, it has been sent out, the maths ones as well. But yeah, this will all get sent out as well. Okay, so the plaques, you can see they're built up on the artery wall. Now imagine if I had a normal coronary artery and there's no plaque on there, there's normal blood flow here. Whereas if you reduce the amount of space in a tube, the pressure is going to be a lot higher, and that's what leads to high blood pressure, and in turn it's going to lead to a heart attack. So essentially high blood pressure is being formed with that plaque being formed, and if that plaque keeps building up, and it will eventually block. If it blocks and there's no blood going through, there's no oxygen going through, so no oxygen going to the cells and no respiration happening, essentially leading to a heart attack. Okay, so it's really important you understand what the build-up can lie up to. Okay, so that's the understanding of the question. It says, which two factors increase the risk of plaque being formed in the coronary artery? Well, a high fat diet definitely increases the risk of plaque. The more fat that you eat, these fatty deposits are going to build up in your arteries as well, even the coronary arteries as well in the heart. Not low blood glucose, not regular exercise, because that's good for it. Smoking can cause that plaque as well. So think about if you've got a cigarette, it's got that tar inside, it's got all them chemicals which essentially can build up on the heart arteries as well, the sticky substance. Taking statins, no. So this last point, statins and stents both help cure this essentially. So what a stent is it's a little it could be like a tube thing, or it could even be like a toothpick thing like this. They essentially stick that inside the artery to open it back up again, in case it's blocked, and it's usually a surgery they do through the arm once somebody's had a heart attack, and they push that stent in and it goes to the coronary artery rather than doing open heart surgery. Statins are a type of medication, so it's thinning the blood, and it's essentially trying to reduce the cholesterol level that's causing that buildup in the arteries as well. So this statins and stents. Regular exercise would help prevent these plaques from building, because you're essentially promoting more respiration from happening. You're promoting from these fats to be not in your body and getting rid of them. Low blood glucose, that's not really relevant to this at all. So that's completely random. Okay, so we've got a 3 marker. So a heart attack can happen when plaque blocks a coronary artery, as we said. So if this fatty deposit plaque keeps developing, it will eventually block it, like so. If you keep eating in the same way and you're not moving and such, and it's just that bad lifestyle. So a heart attack can happen when plaque blocks a coronary artery. During a heart attack, muscle cells in the heart die. Explain how the blockage can lead to the death of muscle cells in the heart. So think about it, the heart is a muscle, and that muscle needs to carry out respiration in order to stay alive. So 3 marks, we need 3 points. So explaining how the blockage can lead to the death of the muscle cells in the heart. Well, first of all, if there's a blockage, there's no blood flow. So you could say less or no blood flow due to the blockage. That's your first mark, to understand that that blockage will cause no blood flow. So if there's no blood flow, the whole point of the heart beating is so oxygen goes around the body. So if there's no blood flow, then there's no oxygen going to the muscle cells in the heart. Always relate it back to context of the question. I see a lot of students also forgetting that as well. Like they'll just say no oxygen, but no oxygen going where? You have to, it's saying here, explain how a blockage can lead to the death of the muscle cells in the heart. So you have to be specific to the muscle cells in the heart. So no oxygen is going to be going to the muscle cells in the heart. So if no oxygen is going there, then there's going to be no respiration at the muscle cells, and that in turn leads to the death. If something can't respire, remember the equation for respiration, always refer back to it. So respiration is you breathe in oxygen, then that reacts with the glucose that you eat from your food. You then breathe out carbon dioxide, and we also release water. And the whole point of this process is to release energy. Majority of things that you do in biology will always go back to this respiration equation. So with the heart, it'll be to do with oxygen. With the endocrine system, it'll be to do with blood glucose concentration. They all affect respiration. Another point to make as well, make sure you say release energy. If you say the word produce, you will lose the mark. Think back to physics. You cannot produce energy. You can't create or destroy energy. It's just transferred, right? So that's why you have to say release energy. And if a cell's not releasing energy, it's going to lead to the death of that cell. Okay, so that's that one. Okay, so this is leading nicely to photosynthesis. So if respiration, is oxygen plus glucose is going to carbon dioxide and water, photosynthesis is just the opposite. So as you can see, so complete the equation for photosynthesis, carbon dioxide plus water. So plant takes in carbon dioxide, we give the plant water, and then in turn using light, so photosynthesis only happens during the day, it doesn't happen at nighttime when it's dark, glucose and oxygen is released from that plant. It says here, what's the chemical formula for glucose? Again, they might not be nice and give you options, you might have to just write it down, but it's C6H12O6. Another thing as well is this, and both of these equations, you need to know the symbol equation for both of these. So I'll write that down. Oxygen, which is O2, plus glucose, which we've just said is C6H12O6, goes to carbon dioxide, CO2, plus water, which is H2O. Remember, with chemistry, you have to balance everything. So you put a 6 here, you put a 6 there, and put a 6 here. I'd recommend memorizing this. Don't try and work out like how to balance it. You've just got 6's in front of them 3, and then your glucose is C6H12O6. So that's for respiration, and photosynthesis is just flip it around. It's the opposite way around. Okay. It says give 2 ways plants use the glucose produced by photosynthesis. So there's a few different answers you can give here. The most obvious one is you should know that animal cells, let me keep it here. Animal cells only carry out, not photosynthesis, respiration. We don't do photosynthesis. We don't have plant cells in us. All animals just carry out respiration, whereas plant cells, they carry out both photosynthesis and, as weird as it is, they also carry out respiration. They do both. So make sure you remember that important distinction between both of them. And remember, photosynthesis happens in the chloroplasts. They might ask you what makes the chloroplast green or the leaf green. It's the chlorophyll that's inside the chloroplast that makes it that. Okay, so back to the question. It says give 2 ways the plants use glucose to produce by photosynthesis. Well, if the plant is also doing respiration, then what it's made from photosynthesis, the glucose, it's going to then use that for respiration. So you could say that it's going to use the glucose for respiration. You could also say that the glucose is converted to starch and stored away. So remember, when so glucose is quite a small molecule, and if you put loads of glucose together, that ends up forming a big molecule called starch. So we can go the opposite way around as well, but putting loads of glucose together, that essentially forms starch. So you could say you could convert it to starch and store it away. So convert it to starch. I'll give you a few other answers that you could have put down. So you could have said to produce fats and oils and store that away. You could also say to produce cellulose. Now remember, cellulose is a type of sugar that's in the cell wall of the plant cells. I don't know why I wrote this. The cell wall. So the cell wall is made up of cellulose. So the glucose can also go to produce that. You could also say to produce amino acids or to produce protein as well. But I think they're the probably 2 safest answers to go with. With science, it's not all about like, is your answer right? Sometimes your answer might be right, but it might not be written like that in the mark scheme. So you really do want to train yourself with what the mark scheme's written like with these questions. Okay, let's move on to this question. Okay, so a person has 2 greenhouses. So a greenhouse is essentially like a glass box, and what it is, it's keeping all the heat inside and the sun directing onto it, containing it within that container to create a really good environment for the plants to grow in. So in each greenhouse, the plants are all the same type, so they're all the same. So essentially, it's like a control variable. They receive the same intensity of light. So intensity of light, how much light, like the strength of the light that they're getting. They're getting the same volume of water as well. So these are all control variables because I guess they're testing something, right? That, and you don't want these factors that affect it. And they're also grown in the same type of soil because they get minerals from that soil as well. So figure 7 shows the greenhouses, and just looking at both these pictures, the only difference is that one's got a wood burner and the other one doesn't. And also these plants are bigger than these ones. So I'm guessing the question's probably going to be along them lines. The person burns logs in the wood burner in greenhouse B. Explain why the plants in greenhouse B grow faster than the plants in greenhouse A for 4 marks. Okay, so we need 4 points for this essentially. So what is that wood burner doing? Essentially, it's increasing the temperature of that greenhouse, right? So that's the first thing you want to say. In greenhouse B, the wood burner is increasing the temperature compared to greenhouse A. So you've got 1 mark for explaining what's the difference. The second mark, now you need to explain, okay, well, what's that going to do? If you've got an increase in temperature, that's going to increase the rate of photosynthesis. It's one of the factors that affect photosynthesis. This is the reason why this is all the same. Intensity of light, the more intensity of light you get, the more photosynthesis that will also happen as well. That's why they've kept it the same. The more water that you're going to get as well, the more photosynthesis is going to happen because the equation is going to keep going. It needs photosynthesis. So you're going to say more photosynthesis takes place. So if more photosynthesis is going to take place, think about the equation for photosynthesis. You're producing more glucose, right? So if you're producing, if you look here, if you're producing more glucose, the oxygen isn't really relevant because the oxygen is getting released off the plant. It doesn't stay with the plant, whereas the glucose stays with the plant. So that's the one that you need to talk about in the products, not the oxygen, which is a common mistake as well by students. You're speaking about the glucose. The glucose is contributing to the growth of that plant. So you would say if more photosynthesis is taking place, then more glucose is being formed. So that's 3 marks. And then for the final mark, you need to explain, okay, so why is that more glucose going to make the plants a lot bigger? A plant's going to go bigger if it's got more proteins and more cellulose made. So you would say something along them lines. So you could say more protein, cellulose formed, so contributing to the growth of the plants. The growth of plants. And that's your 4 marks. So yeah, even this point as well up here, this last point you could have even made for this question over here. The fact that the glucose is stored away to produce cellulose and to produce proteins, which essentially contributes to the growth. So yeah, this 4 mark, I'd say a lot of students got the first mark, but then they struggled to finish the question off and explain why that increase in temperature is really good for the plants growing. So yeah, I would highly recommend the bullet point way of actually answering these questions. If you ended up just writing a paragraph, you might end up writing stuff that's not relevant and then lose marks because you wrote everything that you needed to write, but then you wrote something extra that negates then your mark beforehand. Okay. Another way you could have also answered this question, which some people did, if you think back to chemistry, when you're burning wood, that is a combustion reaction. We're going to cover this in tomorrow's class. That's a combustion reaction, and a combustion reaction is where you get oxygen, you react it with the fuel, so the fuel in the wood burner, you form carbon dioxide and water. So the carbon dioxide also helps with photosynthesis as well. If that wood burner is releasing more carbon dioxide, if you look at the photosynthesis equation, the more carbon dioxide you put into this reaction, then also the more photosynthesis that's also going to happen as well. So you could have also said that as well. So again, yeah, some students did spot that as well. So you could have said the increase in temperature increases photosynthesis. The combustion of the fuel is forming carbon dioxide, which is also increasing photosynthesis as well. So anything along the lines of it's increasing photosynthesis. Okay, question 9, we're on about viruses now as well. So the common cold is caused by a virus. So remember, there's 4 types of pathogens. You've got bacteria, virus, protist, and fungi. It says, give one reason why antibiotics are not used to treat the common cold. Well, antibiotics are only used for bacteria, whereas the common cold is caused by a virus. So you can't use antibiotics for that reason. So you would just say antibiotics do not kill viruses, only bacteria. You could even be more explicit and say the cold is caused by a virus, but it says that in the question anyway. The development of a new antibiotic has many stages. Which is the last stage of the development in the new antibiotic? The last stage of the development is where you actually test it on humans, so you call that clinical testing. Discovery is where you actually discover, okay, I found a new antibiotic. Preclinical is probably using it on mice or other animals before actually using it on humans. And diagnosis, what you're actually using it for, what's the word? What bacterial infection are you actually using that for? Okay, we've got an antibiotic resistant question now. So figure 5 shows the effects of adding antibiotic to a culture of bacteria. So a culture is just one of those petri dishes, and you can see them spots on it and they end up growing. So looking at this petri dish, we can see the ones that are not coloured in, they're the non-resistant bacterium. So bacterium is just referring to one, bacteria is multiple. Then in the second one, after the antibiotics added, you can see the colour one remained, and the coloured one is the resistant bacterium, so the one that's resistant to the antibiotic working on them. And then after, you can see it multiplies, so that person's essentially going to, if this was in a person, that person would become ill again after taking the antibiotics because there's one resistant bacterium in there that's replicated. So it says, explain how antibiotic resistant bacteria have evolved and use information from figure 5. So you can't just generically speak about antibiotic resistance, always refer back to the question. Okay, so for 4 marks, we're going to say that some of these bacteria have a mutation inside, which is causing them to have that resistant gene, which is basically this one. It's mutated. So you would say some bacteria have a mutation. So a mutation is just a change in the DNA sequence. So therefore it's become resistant. That's why it's essentially changed. It become resistant to the actual antibiotic. And then after the antibiotics added, you can see that the non-resistant bacteria is killed. So after antibiotic added, the non-resistant bacteria is killed, which we can see, and the resistant one remains basically. So resistant bacteria remains, and when that resistant bacteria remains, there's no competition for this resistant bacteria. Like, it doesn't need to share its resources with anything else. So because there's reduced competition, it's going to reproduce. So there's no competition for this because there's nothing else competing for what it needs to essentially reproduce. So no competition, so therefore it reproduces. And once it reproduces, you can see this is what's happened over here. So for the final mark, you would just say all bacteria are antibiotic resistance because they have the mutation. They all have the mutation. So yeah, that's 4 marks, 4 points. Again, some of this seems a bit redundant to say because it seems obvious to write it down. So some students hold off from saying it because they'll say, well, it looks obvious that the resistant bacterium's reproducing because it shows it in the picture, so they don't write it down. But essentially, a lot of these questions in science is just write what is in the question again whilst explaining it, and you've got the mark. Okay, so 4 points, 4 marks. Okay, so pepsin and trypsin are enzymes that break down proteins. Now remember, enzymes are biological catalysts. They speed up a reaction without getting used up. So the analogy that I use, imagine you're making a cup of tea, you're putting sugar in the tea, you're not just going to leave it. You use a spoon, which is a catalyst, and you're going to mix it in order to dissolve that sugar faster. But the spoon's not melted, you can use it again. So it's a catalyst, speeds up a reaction without getting used up. So pepsin and trypsin are enzymes that break down proteins. Now, in the exam, they can give you any catalyst that you've never seen before, but just apply the knowledge that you already know about how pH and temperature affects enzymes. And in this question, we're speaking about pH, so you should recognize these bell-shaped graphs are always when they're speaking about pH. If they're speaking about temperature, what happens with temperature is, so I'll do enzyme activity. That looks terrible. 2 seconds. So enzyme activity, but instead of having pH on the x-axis, I'm just going to put temperature. You should recognize the shape of these graphs. Now, what happens is if you increase the temperature, the enzyme activity will increase, but then eventually you'll get to a point where it just drops down like that. So hold on. So increase, increases, increase, and then it just drops straight down to 0. So this here, if it was in a human, it would be 37 degrees because 37 degrees is our optimum temperature, the temperature we work best at. After that temperature, the enzyme has denatured. Now, when we speak about the enzyme denaturing, imagine the enzyme look like this and the substrate's going to fit inside the active site. The enzyme denaturing means the active site has changed shape, so it's not going to fit anymore. So you call it denatured. You have to use that word. Denatured just means the active site has changed shape, therefore it's not working anymore. Okay, if you've got any questions, you can put them in the chat as well as we're going along. Okay, so we've got figure 8 shows the results of an investigation in the activity of pepsin and trypsin at different pH levels. So we can see pepsin works at acidic levels. So remember, a lower pH is acidic, and trypsin works at a higher pH level. That's more alkaline, basic, essentially. Okay, so they work in different conditions. So if I was to think where is acidic in the body, that's the stomach. We've got hydrochloric acid. So we find pepsin inside the stomach. I think it's a type of protease, but you don't need to memorize that. Which molecules are produced when a protein is broken down? So proteins are broken down into amino acids. Fatty acids are and glycerol is what lipids break down into, and then starch breaks down into sugars. Starches are big molecules as well, so it breaks down into that. Okay, let's move on. Describe the trend in the graph for the enzyme pepsin. Use data from the graph to support your answer. So for 3 marks, explain pepsin essentially. So we can see, let's describe the trend first. From, if we look at that, it doesn't start at 0, it starts over here. So between, that's 0, 1, 2, 3, 4, 5. So we've got 5 boxes between 0 and 1. So if I do 1 divided by 5, that's 0.2. Each box is worth 0.2. So that there is 0.2. So what you could say is from 0.2 to this peak over here, which is, that's 2, right? It's increasing. So to pH 2, say it is pH, actually, you shouldn't just write, because what is 0.2? I'm not given context as to what that is. So from pH 0.2 to 2, you can see the enzyme activity increases. So you just described what's happening initially. For the next one, you could say that the optimum of this enzyme activity, so the best is at that 2. So the optimum is at pH 2. So that's where it works the best, so optimum enzyme activity. And then finally, just describe the other part of the pepsin. So from 2 to that point there, let's say 3.6. So from 2 to from pH 2 to 3.6, the enzyme activity decreases. And that's your 3 marks. So you've just described the thing. So be careful when it says describe, you're just saying what's on the graph. If the question said explain, you're explaining why certain parts are a certain shape. So be careful of that. If you had explained each part, you would have lost the marks because your answer's right, but you've not answered the question. You explained it and not described it. It says here, state the optimum pH for the enzyme trypsin. So trypsin is this one, and the optimum is over here at 8. Okay, so we'll just write pH 8. It says explain why there's no trypsin activity at pH 5. So that's pH 5. pH 5 is over here, so why does trypsin not work there at all? Well, 5, if you think about the pH scale, that's very acidic. Trypsin does not work in acidic conditions. It's working in more alkaline conditions. So you would say pH 5 is too acidic. pH 5 is too acidic for trypsin. And then you need to explain, because the question says explain why is that an issue for trypsin. So this is where you're speaking about the enzyme active site and the denaturing. So you would say at pH 5, the active site would change shape. And if the active site changes shape, the substrate will not fit into the enzyme. And if the enzymes, the substrate's not going to fit into the enzyme, you call the enzyme denatured. So that's your final mark. Enzyme has denatured at pH 5. That's why there's no enzyme activity there at all. So yeah, that's 3 marks there. Okay, it then says temperature is a variable that should be controlled in this investigation. The reason why we said the temperature should be controlled is because temperature, as we spoke about here, is also a factor which affects enzymes. So the 2 factors you need to know for GCSE for enzymes is pH and temperature. They're the only ones that they expect you to know. To give one way that the temperature could be controlled, you could use a water bath. So a water bath. So do the experiment in a water bath just to keep that temperature the same. Okay, so malaria is a disease that causes damage to the blood and liver. Which type of pathogen causes malaria? You should know that's a protist. So remember, the mosquito carries a protist, but the mosquito isn't actually the protist. It's a vector that carries it. So it says here, state how the pathogen that causes malaria is spread. Well, it's spread through the vector mosquitoes, essentially, by vectors. And these vectors are the mosquitoes. Yeah, so that's how it's spread. So it's essentially spread through the blood. So then the mosquito would essentially bite whoever is passing it onto, and then push that protist inside. The protist, it gets it from swamps and wet areas. That's why there's not a malaria in the UK, because we don't really have any swamps here. But when you go abroad, there does tend to be swamps and such where it gets it from. Okay, one other thing as well that I hope you've noticed as well is there's a lot of figures and graphs within the biology papers, especially as well, where they want you to interpret, work out the mean, work out percentage changes and such as well. So you need to be quite comfortable with them math skills as well when it comes to these biology papers, and interpreting charts and graphs and understanding what the question's asking as well. So measles is a disease caused by a virus. Figure 9 shows the number of measles cases reported in England and Wales from 1985 to 2015. So if you look, as the years are increasing, the number of measles cases reported is decreasing. So it's getting lower as time's going on. So for part B, it's saying, explain one conclusion that can be made about the change in number of the measles cases reported from 1985 to 2015. So the first things first is we notice that as the years are increasing, the number of measles cases reported has decreased. So from 1985, be specific with the numbers, to 2015, the number of measles cases reported has decreased. There's your first mark. You've just described what's seen. Now you need to explain it. Why do you think it has decreased? Because it's an explain question, not a describe. Well, measles, think about it. We get when babies are born in the UK, we get the MMR vaccine, so the measles, mumps, and rubella. So we get vaccines, well, if you opt into it, but majority do. When you get that vaccine, you're essentially not going to get measles because you're immune to it. So you would just say, because people have been immunized, more people are immune to the disease. So more people are immune. And the reason why we're immune is because of the vaccine. So you could even write that down as well if you wanted to, due to the vaccine that's essentially being given out. So if you just want information on that, you should be aware of the MMR vaccine that kids tend to have when they're born here. And if you didn't have that, then you're susceptible to get measles, mumps, and rubella, and it would kill you. There's no way to cure that. That's the reason why they try and push you to have this vaccine. Okay, so describe 2 ways the immune system will respond to an infection by a pathogen. So white blood cells essentially kill pathogens, right? So we can start with that. So white blood cells destroy, or you could say kill, pathogens that enter in. That's their job. So you could even be specific with that. You could even speak about phagocytosis and say that it engulfs pathogens. You would have got the mark for that as well. Another way white blood cells also destroy pathogens is by producing antibodies or antitoxins. So you could speak about that as well. So white blood cells can produce antitoxins or antibodies. I would say just say one or the other, don't say both. You can even talk about the memory lymphocytes as well, which basically means that when it comes again, your bodies have the memory of the antibodies already produced and it's already ready to basically kick in as well. So you could say that as well. There's quite a few things that you could say here. Just going to write phagocytosis here in case you want to speak about the engulfing. So you just know you could also speak about that as well. I don't even know how to pronounce that. Beriberi. Okay. Beriberi is a disease caused by lack of vitamin B1 in the diet. Give one reason why beriberi is classed as a non-communicable disease. Now, non-communicable means that it's not caused by a pathogen. You can't pass it to somebody. So another example of a non-communicable disease is cancer. If somebody's got cancer, just by you sitting with them, you won't get cancer because it's not caused by a pathogen. Whereas a communicable disease is caused by pathogens. So if somebody's got a cold, which is caused by a virus, if you're with them, you could potentially catch that cold because they could pass the virus on to you. So that's a communicable one. So all you would just say is give one reason why beriberi is classed as a non-communicable disease. You would say beriberi is not spread from person to person. So essentially not caused by a pathogen. It's caused by lack of B1 in the diet, right? So that's not spreading from person to person. So yeah, again, another thing you should notice whilst we're going through these questions as well, they're going to throw really random things at you you've never seen before, like beriberi. When you sit your GCSEs this year and anyone sitting here next year, you're going to see things in that science paper that you've never seen before. But the answers are stuff that you have already learned. You just need to apply it to the question. That's the skill that comes with the science papers. Okay, moving on. Just wary of time, and we're only on question 11. Okay, we should finish it. Okay, beriberi can affect reflexes. So figure 10 shows a reflex arc. So you can see we've got 3 neurons here. You should immediately, as soon as you see them 3 neurons, sensory, relay, and motor neuron is what you should be thinking. Skin of the finger, the receptor is going to detect that pin. That's essentially going to hit that skin. The skin's going to detect that. It's going to pass through the neurons, and then the muscle in the arm, I'm guessing, would pull you away from the pin, right? But beriberi is going to affect these reflexes. So when the skin is pricked by a pin, electrical impulses travel through a reflex arc, which you should be aware of. Describe the path taken by electrical impulses from the skin to the muscles in the arm. Include the name of the neurons X, Y, and Z in your answer for 6 marks. So a 6-mark question, just explaining the nervous system, and you just have to use the example given to you over here. Okay, so I think it's a pretty nice 6-marker. So just going to generically summarize it, but obviously you'd have to explain it a bit more. So the electrical impulses, before speaking about that, let's speak about the receptors actually. So at first, if you look here, the skin is pricked by the pin, and the skin is the receptor. So that's the first stage in this reflex arc. The skin is a receptor, and the pin is a stimulus, the stimulus which is actually being detected by the receptor. So the pin, that's the stimulus. Obviously, write this in full sentences. Just for time, I'm just kind of analyzing it. Okay, next, once it's detected by the receptor, it's passed to neuron X, then neuron Y, and then neuron Z. So neuron X, then neuron Y, and then neuron Z. And the order, which you should remember, is the sensory neuron first in that stage. And then after that, it goes to the relay neuron, which relays it to the muscle cells from the spinal cord in the brain, to the motor neuron, which then goes to the muscles, in this case, not the glands, to essentially move your hand. And in between each of these neurons, you could even mention this as well, this is a 6 marker, so you want to just put everything to do with the reflex arc and the nervous system that you know. So in between each of these neurons, we've got synapses, so the gaps which have chemicals inside to essentially pass on that electrical signal. So you can see these gaps here, there's a gap that passes through each other. So you can speak about that as well. Then the chemical transmitters are passed through that synapse. Chemical neurotransmitters, which pass it through that. And then the last stage is the muscle. Now the muscle is known as the effector. Now, one thing as well you don't want to do as well, again, and I'm reiterating, but you always have to give it to the context of the question. A lot of people will just end up writing everything that they know about the nervous system. So for example, knowing that the nervous system affects both muscles and glands. Glands are not relevant here. We're speaking about the muscle in the arm. So why would you say the effector's a gland when it's not relevant in this question? So you would say the muscle is the effector. And what's the effect of that muscle? Well, the muscle is going to contract because it's going to move away from the pin. So then you would say that contracts. And essentially that's for the safety of the pin. And you would say that this response is also very fast. You wouldn't want it to be slow, right? You wouldn't want the pin to go fully further in and this whole process is going very slow. So it's just really fast. And that's why it's known as a reflex arc. So just some keywords I want to highlight. So receptor, stimulus, each of these neurons, the synapse, talking about the transmitters, and also the fact that it's an effector as well, so the reflex arc, essentially. Okay, let's move on, just a bit wary of time. Okay, drinks contain different substances. A drinks company claims that a drink contains sugar, protein, and fat. Glucose and fructose are different sugars. Notice usually when the end in ose is a type of sugar, like even cellulose that we spoke about before ends in ose, it's a type of sugar. Fructose has a much sweeter taste than glucose. Now, anyone that has drank like coconut water has sometimes has added fructose inside it as well to make it sweeter. So if you look on like a packet of coconut water, you'll see fructose also written there as well. Now, the reason why fructose is used rather than glucose is because if you put glucose in, glucose will help contribute to respiration, so good for energy. So stuff like Red Bull, Lucozade, all these energy drinks have added glucose inside to help you with respiration, but you don't always want that. So it says here, so fructose has a much sweeter taste than glucose. Suggest, so we've got a suggest question again. Suggest 2 reasons why the drinks company uses fructose in drinks rather than glucose, and do not refer to sweetness in the answer. So kind of just what I said, if fructose is sweeter, then you would need less of it than glucose to put in, right? To match the sweetness levels, more glucose would be needed than fructose, so you could say that first. So less fructose needed to be used than glucose, because that's the only reason they're adding it in. They're adding it to change the sweetness of the drink. So less fructose needed, so that's like an economical benefit for that. You could also say the whole energy thing that I was speaking about, so less energy in the drink when using fructose. So using the coconut water example for example, you wouldn't want coconut water to act as an energy drink. So if companies started putting glucose in coconut water, you've turned it into an energy drink, then it's kind of lost its purpose. So that's the reason why they use fructose there as well. Okay, yeah, so these suggest questions are quite tricky because it's kind of just basing your general knowledge, kind of. Okay, describe how a student could test for the drink of sugar. So you need to know your color tests. When you're testing for sugar, this is Benedict's that you're using. So Benedict's solution is 3 marks. So what do you have to do with the Benedict's? You have to heat it. So you heat the solution, and it says color of a positive result. So a positive result means what's going to be the color of that Benedict if there is sugar inside it, and it would be brick red. is how they describe it. You could just say red as well. You don't have to say brick, but that's usually what is said in textbooks. So it changes to brick red. Describe how a student could test for protein. The protein test is the BIRET. The BIRET reagent that you use. There's just 2 marks. The reason why the other one was 3 marks is because you had to say heat solution. So the color of a positive result, if there is protein, the BIRET is purple. Okay, so you need to know both the negative and the positive sugars and proteins. Okay, so the human digestive, got 6 marker here as well. The human digestive system breaks down proteins and fat in the drink. Describe how protein and fat are digested. You should include the enzymes involved and where the enzymes are produced. So for 6 marks. So you need to also know what they break down into. So we'll start off with the protein first. I'll put some bullet points. But with these 6 markers, I would highly recommend writing them in actual sentence structure. The other ones, it's okay to use bullet points, but the 6 markers, do write them in a good English format, if that makes sense, because they give you marks on the way you articulate your ideas as well. So protein is broken down into amino acids. And the enzyme that's used for this is proteases. Are used to break down protein into amino acids. Now, proteases, you just need to speak about where they're found. Well, they're found in the stomach. The stomach is quite acidic. It's also produced in the pancreas as well. So these are where they're produced. The stomach and the pancreas is also produced in the small intestine as well, if you want to write that down. Remember, the small intestine is where things are also absorbed in as well. You could also speak about how the stomach has hydrochloric acid inside. So the proteases that are found are adapted for the pH of this area, which is really acidic. You could speak about that as well. Okay, so that's for proteins. Let's speak about fats now. So it's probably a good split, like 3 marks each. Now with fats, they're broken down into fatty acids and glycerol. And the enzyme that's used for this is lipase. So remember, fats are essentially just another word for lipids as well. So lipids and fats are the same thing. Okay, so broken down into fatty acids and glycerol. Talk about where they're produced. So lipase is produced in the pancreas. It's not produced in the stomach. The proteases are produced in the stomach. It's also produced in the small intestine. So these are where they're produced. You could also speak about bile as well. So the reason why you would speak about bile is because it emulsifies fats, and fats is relevant here because it's speaking about fats. So in the digestive system, the biles, which are produced by the liver, emulsifies the fats. And the reason why it emulsifies it is basically to give it a large surface area. So write LSA, so large surface area, which tends to, again, that also comes up quite a lot in biology as well, stuff like with the alveoli, and other things as well, catalysts and so on as well in chemistry. Okay, moving on. So figure 1 shows a light microscope. These are the ones that you just use in school. Draw one straight line from each part of the microscope to its function. So part S, that's the rotating dial. So the rotating dial, that's to focus the image. So just be that one there. So when you're actually looking at the specimen, you're rolling that just to focus it, so you don't want it to be blurry. You're just trying to adjust that. Part T, they're the magnification lens. So you can see there's different lenses that you would use. So that's to magnify the image. So that's there. To hold the slide in place, that would be the stage. So the stage that's over here. To provide light to see the image, that would be down there. To stain the cells, that's completely different, not even part of the microscope. You do that before the microscope. Okay, we've already kind of spoken about this. So state one advantage of using light microscope and one advantage of using electron microscopes. Now, a light microscope is cheap, so I'm going to list a few reasons, but obviously just choose one. So you could say the light microscope is cheap. It's a lot easier to use than an electron one. Okay, so you could say one of these, so cheap or easier to use. An electron microscope, we need to put an advantage down. So I was thinking expensive, that's not an advantage, but you could say it's a higher resolution. A higher resolution, so you get more detail. Had they asked about the pros and cons, the cons of using a light microscope is You wouldn't get a high resolution. An electron microscope, the specimen has to be dead. It's also a lot more expensive to use an electron microscope as well. Okay, let's move on. Insulin is a hormone that controls blood glucose concentration. So remember, hormones are chemical messengers. Are they here? So chemical messengers. Which endocrine gland produces insulin? That's the pancreas. Pancreas, the thyroid gland produces thyroxine. The adrenal gland produces adrenaline, the hormone adrenaline. The pituitary gland, that's the one that's just behind the brain, and the master gland, and they produce other extras like LH, FSH. The one for thyroxine, is it TSH, something like that? But that comes from the pituitary gland. State how insulin is transported from its endocrine gland to its target organs. It goes through the blood to where it needs to go. Name the main target organ for insulin. That's the liver. And the reason why it's the liver is because insulin is used to lower the blood glucose concentration. So to do that, it's going to tell the body use the glucose for respiration. But if it's used everything that it needs to use for respiration and doesn't need any more, the insulin tells the glucose to go to the liver and store it away as glycogen. So that's the reason why it's a target organ for insulin. A lot of people get confused with that, like why the liver? It's because it's to store away for glucose as glycogen if the blood glucose concentration is too high. Okay, people with type 2 diabetes have cells that do not respond to insulin. State 2 ways that people with type 2 diabetes can control their blood glucose concentration. Well, essentially reduce the amount of starch and carbohydrates that they're taking. So carbs and starch in their diet, because that's what's essentially promoting more glucose in your body that you can't get rid of. Another thing you could do is exercise. You could, as a third point, if it was a 3 marker, you could say lose weight as well. So people with type 2 diabetes tend to have quite a big waist, which also contributes to it as well. So losing weight would help people with type 2 diabetes. It wouldn't work with type 1. Type 1 diabetes is where you're not producing any insulin at all. Whereas with type 2, you are producing insulin, but your body's not responding to that insulin. Okay, we've got 15 minutes left, so I'm just going to quicken up a little bit. Cystic fibrosis is a genetic disorder. Now, what that means is it's caused by your parents, essentially. You don't get it from here. It's a gene that you carry down. So it affects the movement of substances into and out of the cells. Which part of the cell controls the movement of substance in and out of the cell? That's the cell membrane. That lets certain things in and out of the cell. And mitochondria is for respiration. That's what that happens. So cystic fibrosis is caused by recessive allele lowercase b. What's the name given to allele B? That's dominant. So capital is dominant, lowercase is recessive. Which term describes the genotype? Capital B, lower b, that's heterozygous. And homozygous is if they both were the same. So homozygous is if it was capital B, or lowercase b, lowercase b. So hetero is where it's 2 different ones. Okay, phenotype is basically what you're displaying. Chromosome is essentially DNA coiled up. So it's not them. Okay, 2 people plan to have a child. Both people have the genotype capital B, small b. So I guess I'd have small b here, right? We've got a Punnett square here. Complete the figure. So you can see here capital B is already here. For this one here, you'd have capital B, small b. Now, you never ever get something like that. You never write the lowercase letter first. It always goes uppercase and then lowercase. Over here, we've got capital B, small b. And finally, we've got small b, small b. Now, what is the chance of this child will have cystic fibrosis? Now, it's a recessive disorder, so you have to have both of them lowercase, which is just this one here. So that's 1 out of 4 chances. So there's 4 options. So 1 out of 4 as a percentage is 25%. You just tick that. An embryo can be tested to find out its genotype. What's the name of this testing process? That's just screening. Selective breeding is what we spoke about earlier with them chickens. Genetic engineering is where you're like making a GM crops. So for example, like all apples kind of look the same, or because you're genetically modifying them, for example. So yeah, screening is that process. Inherited disorders can be caused by changes in the DNA. What is the name of the change in DNA? We kind of spoke about this before with the antibiotic resistant one. So it's a mutation which causes a change in DNA. Genome is what you're made up of. Helix is the double helix structure with DNA. So like so. Inheritance is one cause of variation in a population. Environmental factors also cause variation in a population. So what's basically your surroundings? So why are we like different compared to somebody that's living, I don't know, in Egypt, for example? Like why is it different? That's the environment. So suggest one environmental cause of variation in the human population. Do not refer to inheritance or changes to the DNA in your answer. Well, there's quite a few different things that could be different. So your diet could be different depending on where you are. Your behaviors. So for example, you might have like tattoos. For example, if you're in a certain place, you might dye your hair a certain color or so on. Environmental causes such as like variation in light. So we don't get a lot of sunlight here, but other places in the world get a lot of light. So that would also be different as well. So I'm just going to put a few here and you would just choose from that. So diet, something along the lines of diet, behavior, or something to do with the environment like sun. Or this could also be to do with like tattoos or something like that. So just name one environmental factor. Okay, so figure 2 shows organs and glands in a human body. Label the glands. Okay, so you've got the brain here. So just with the brain, obviously we spoke about this endocrine system they're speaking about in this question. Or endocrine, however you pronounce it. So this is the pituitary gland. Pituitary gland just sat underneath the brain. We've got the pancreas there, we've got the kidneys. These are the adrenal glands, which are sat just behind. Adrenal glands. And then over here, this is a female, so that would just be the ovaries. Obviously, a male does not have ovaries. I'm just going to write ovary because it's pointing to 1. Okay. Cells in the pancreas detect blood glucose concentration. What type of cells detect blood glucose concentration? That would be receptor cells. So remember, with the receptors, it affects both muscles and glands. So receptor cells detect the blood glucose concentration. The pancreas produces insulin. How is insulin transported from the pancreas to the rest of the body? We already had a question like this. The reason why I put it out twice was because this is from AQA and the other one was from EdXL. So I just wanted to show you the similarities between the papers. More or less, they're exactly the same. So yeah, this is the blood, as we spoke about before. Which organ's a target organ for insulin? We've already answered this. It's the liver, because that's where it stores it. Which chemical is a store of glucose in the human cells? Loads of glucose in the human cells is stored away as glycogen. Okay, cellulose is what's in the plant cell walls. Proteins are, well, they're just proteins, right? Okay, let's speed through these last few questions. We've got a few more. Students investigated the effect of the number of hours of sleep on reaction time. Now, just thinking about that logically, the less sleep you're going to have, the slower your reaction time is going to be. So that's a bit obvious. So that's kind of what you're expecting. The students used the reaction test card shown in figure 4 on I haven't put it, but it was essentially they had to just give a rating. Basically, it's not really relevant to this question anyway, but they use a reaction card test. They've got the method. So there's student A, student B, and there's the reaction test card they're using. So there's a method used. Okay, now this is testing your control variable, dependent variable, and independent variable. One is so controlling, keeping the same, dependent what you're measuring, independent is what you're changing. So looking at this, so recording the number of hours of sleep that student A had, student B holds the lower edge of the card. Student A holds their thumb and forefinger slightly apart. Student B drops the card. Student A then catches the card as quickly as possible and record the number shown at the top of the number and repeat steps 1 to 6 with other students. So what you're going to control, so basically keep the same. So number nearest the top of the thumb, no. Number of hours of sleep, we're not keeping that the same because that's what we're investigating. Number of students tested, that's not a control. Start with the lower edge of the card level with the thumb. Yep, that's a control variable because everyone's starting at the same point. Okay, so the independent variable, that's what you're changing. So we'll do that one next. So this is change, this is measure, and that's what you're keeping the same. So if you want to remember what each of these variables mean, it comes up in every biology paper. Okay, so we'll do independent variable. What we're changing, we already spoke about that. The number of hours of sleep is what we're changing because that's what we're testing on. How is that affecting? The dependent variable, so what we're measuring, we're not measuring the number of students tested. We're measuring the number nearest the top of the thumb when the student catches a card, so what number they last land on. Okay, so motor neurons are involved in reactions. Figure 7 shows a motor neuron. Okay, so there's the muscle cells and then there's the actual neuron. It says explain one way the motor neuron is adapted to its function. So there's a few different things that you could say, but just looking off the bat of that, it's quite a long cell, right? So that's 1 mark. So it's a long cell. Well, how is that gonna, we need to explain why is that adapted for its function being long. It's a bit like that first question we did, how's the flagella adapted? Is for movement, right? So long cell, because what's the function of the motor neuron cell? You explained that to transport information. It's essentially a signal, right? The motor neuron, the electrical impulses that it's transporting. Now, where is it transporting it from? So it's going from the central nervous system to the effector. And that's your 3 marks. There's other things you could have said as well. So you could have talked about the many branches that it's got. So these here, which is the same reason. You could have said it's attached to the muscle cell. That's an adaptation as well. You could have said many mitochondria, but I can't see that on the diagram anyway, so I probably wouldn't say that. You could say insulation or fatty sheath as well. So yeah, you could say any of them, but I would say go for the most obvious, which you can see is it's a long cell. Okay, question 18. In humans, gas exchange often occurs in the alveoli of the lungs. So here we've got the alveoli, this here, and then this is the capillary that's surrounding it. And as you can see, oxygen is going out, and then carbon dioxide is coming in to the alveolus, which is 1. Alveoli is many. Which process moves carbon dioxide from the blood into the alveolus? That's just diffusion. State 1 adaptation of the alveolus that increases the rate of gas exchange. So there's quite a few things you could say. You could say the walls are thin, which you can see. So if the walls are thin, then diffusion can happen, right? So you could also say, I'm going to put a few others here as well. You could have said large surface area, which comes up quite often, or you could say a good blood supply, because you need the blood constantly moving through the capillary so we can keep the oxygen and carbon dioxide moving. If it just stayed still, that wouldn't happen. So choose one of the 3. Describe how blood is moved from the heart to the lungs. Okay, so you're describing part of the circulatory system where it's going from the heart to the lungs. So the heart is essentially contracting. That's how it's actually pumping the blood around. So the heart's contracting, so it pumps blood. For your first mark, and you would say it would go through, like, where is it going through? And that's going through the pulmonary, because pulmonary is to do with the lungs. And what vessel is taking blood away from the heart? That's artery. Vein is taking it to the heart, artery is taking it away. So through the pulmonary artery. You could even say under pressure as well, it's going. You don't have to say that for the mark though as well. Okay, last few questions. I'm going to whiz through these ones. Okay, the heart pumps blood. Explain why the wall of the left ventricle of the heart is thicker than the wall of the right ventricle. You'll think about it on the left ventricle. Remember, with the heart, it's always the opposite way around. So if I was to draw a heart, this is not the right side, this is the left side. Let me just write left. And this is the right, so it's the opposite because you're looking at it as though it's a person in front of you. So it's saying, why is the left ventricle of the heart thicker? Well, think about it. That left part is pumping blood to the rest of the body. So if it's pumping blood to the rest of the body, it's doing that at a higher pressure. So if it's doing that at a higher pressure, then it needs that thicker wall. So there are your 2 marks. A centrifuge can be used to separate different parts of the human blood. If anyone's seen Jurassic Park, it's them like circle things and you spin them essentially to separate out. Name part X, that's just the plasma that's on top. That's separated out from the blood. And you know that because it says the blood is separated, and we know the blood is made up of 4 things. There's the white blood cells, there's the platelets and red blood cells. So part X must be the plasma that's separated out. Which substance needed for cellular respiration? That's carried by the red blood cells. That's just the oxygen. Name 2 types of white blood cells, so phagocytes and lymphocytes. I'm gonna just rush through this last part. Okay, when a person donates blood, 470 centimeters cubed of blood is removed from the body. Red blood cells make up 44% of the volume of blood. Calculate the volume of the red blood cells in 470 centimeters cubed of donated blood. For 3 marks, this is a steal of marks. All you're doing is 44% of 470. So putting that in the calculator, 44% is 0.44 of, just times it by 470, and you end up getting 207. That's insane that they give 3 marks for some of these maths questions because they're quite straightforward. Before donating blood, a person has a small blood sample taken to check that their blood is healthy. State 2 precautions a doctor should take when collecting this sample. Well, you're dealing with blood, so you probably should be wearing gloves. That's one point you could have said. You would sterilize the needle that you're using. Sterilize the needle. You wouldn't want to put a dirty needle into somebody as well. You could have even said something like, once you've taken the blood out, cover that wound so it's not open as well to pathogens getting inside because it's punctured skin. Okay, I'm probably just going to take 2 more minutes extra. I just want to finish these last 2 6 markers, if that's okay. So the arrows in figure 11 show the direction of water movement through a tree. So we can see water's moving inside the roots of a tree. It's going up the tree, and it's also coming out as well of the leaves that's on top of the tree. Explain how water's moved from the soil through the plant and into the air. So we just described the process for 6 marks. So we're speaking about each section. So let's speak about the roots first. So how is water moving through the roots? Well, water enters the roots. The reason why water enters the roots, think about it, when you're watering a plant, where do you put water? You put water into the soil. So it's going from the soil into the roots. You could talk about the root hair cells. Cells for that one. Talk about how they've got a large surface area. Again, that reason has come up again because we're explaining, don't just state, you have to explain why it's taking it in. How does it actually take it in? Well, it's movement of water, so the process is osmosis. Osmosis is just diffusion, but with water particles through a semi-permeable membrane. Okay, so that's the roots. The next part it's going is the stem or the trunk of that tree. Now through the stem, remember there's a xylem and phloem inside the stem. The xylem is for water, phloem is for food and nutrients. So we're talking about the xylem here. And remember the xylem, it just goes in one direction. So it's the long, thin, the water's being pulled up. Pulled up. So yeah, it just goes in one direction. Is probably what I would say as well. They call it something, but I forgot what it's called. And then after that, it's the leaves. Where it's going to now, it's escaping the leaves. So first, it's getting into the leaves by osmosis. Is how it's getting into the leaves in the first place, from the xylem into the cells. And then you would talk about how water evaporates from here. So water evaporates from the leaves, and you should know that this process is called transpiration. So you're speaking a little bit about transpiration here, water leaving the leaves. You could also speak about the stomata as well. So that's on the leaves. The stomata, when they open on the bottom with the guard cells, you could talk about all that process as well, because that also contributes to water leaving as well. Yeah, you're just explaining all that for 6 marks, essentially. And finally, there's the last question. Scientists have collected and stored seeds from species in the same genus as potatoes. In the future, these seeds may be used for genetic modification of potato plants. So this is a GM question. Genetically modified potato plants could help the supply food to the human population as the climate changes. Explain why genetic modification of crop plants may be important for human population to survive climate change. So your first few marks comes from speaking a little bit about climate change. what's happening with climate change, the change in weather patterns, and what can that cause? It can cause flooding, so that would in turn affect like these plants being caused. So if flooding happens, and there would be reduced farming land. So therefore, crops that are currently there wouldn't survive. So you could speak a little bit about climate change first, and then after you're going to speak about the benefits of GM crops and how they form. So GM may allow crops to grow in a wider range of conditions because you're selecting the best ones. You might make them resistant to certain diseases with climate change that is coming across. So you would pick the best ones and then you keep modifying them. You can store them for longer. You might increase the crop yield. So therefore, that would increase the increasing human population. And then you would talk about variation as well, so how you would actually breed them. So you could speak about selective breeding, asexual reproduction because it's just a plant, and then the rate of evolution and natural selection you'd also speak about as well. So these, I would say these ones are the more trickier ones to speak about because it's easy to go off on a tangent. But with these 6 markers, you kind of showing off all the information that you do know for biology. So that's the biology class. I hope that was helpful. I'll send out all these exam questions. The recording will also get sent out as well. If you've got any questions, you can drop them in the chat. But if not, I will see you tomorrow, same time for chemistry as well. And I'll get that sent out. Okay, thank you for joining.